Hello, welcome to Biology, the scientific study of life. Signal amplification, elaborate enzyme cascade amplify the cell's response to a signal at each catalytic step in the cascade. The number of activated products is much greater than in the preceding step. For example, in the epinephrine triggered pathway in this figure, each adenylyl cyclase molecules catalyze the formation of many C, A, and P molecules. Each molecule of protein kinase phosphorylates many molecules of the next kinase in the pathway, and so on. The amplification effect stems from the fact that these proteins persist in the active form long enough to process numerous molecules of substrate before they become inactive again. As a result of the signal's amplification, a small number of epinephrine molecules binding to receptors on the surface of a liver cell or muscle cell can lead to the release of hundreds or of millions of glucose molecules from glycogen. Specificity of cell signaling. Consider two different cells in your body, a liver cell and a heart muscle cell. For example, both are in contact with your bloodstream and are therefore constantly exposed in many different hormone molecules, as well as to local regulators secreted by nearby cells. Yet the liver cell responds to some signal but ignores others. And the same is true for the heart cell, and some kinds of signals trigger responses in both cells, but different responses. For instance, epinephrine stimulates the liver cell and to break down glycogen, but the main response of the heart cell to epinephrine is contraction, leading to a more rapid heartbeat. How do we account for this difference? The explanation for the specificity exhibited in cellular responses to signals is the same as the basic explanation for virtually all differences between cells. Different kinds of cells have different collection of proteins. Again, more notice the steps also: cells A, cells B, or cell B, cell C, cell D. Notice this figure. The response of a particular cell to a signal depends on its particular collection of signal receptor proteins, relay proteins, and proteins needed to carry out the response. A liver cell, for example, is poised to respond appropriately to epinephrine by having the proteins listed in this figure, as well as those needed to manufacture glycogen. Those two cells that respond differently to the same signal differ in one or more of the proteins that handle and respond to the signal. Notice that different pathways may have some molecules in common. For example, cell A and B and C all use the same receptor protein for the orange signal molecule. Differences in other proteins account for their different responses. In cell D, notice that the last one, a different receptor protein is used for the same signal molecule, leading to yet another response. In cell B, for instance, a bath that is triggered by a single kind of signal diverges to produce two responses. Such branched pathways often involve receptor tyrosine kinases, which can active multiple relay proteins, or second messengers, which can regulate numerous proteins. Essentially, two pathways triggered by separate signal or converge to modulate a single response, branching of pathways and crosstalk interaction between pathways are important in regulating and coordinating a cell's response to information coming in from different sources in the body. Moreover, the use of some of the same proteins in more than one pathway allows the cell to economize the number of different proteins it must make. Signaling efficiency, scaffolding proteins and signaling complexes. The signaling pathway in this back to this picture are greatly simplified. The diagrams show only a few relay molecules, and for clarity's sake, display these molecules spread out in the cytosol. If this were true in the cell, signaling pathways would operate very inefficiently, because most relay molecules are proteins, and proteins are too large to diffuse quickly through the viscous cytosol. How does a particular protein kinase, for instance, find its substrate? Recent research suggests that the efficiency of signal transduction may in many cases be increased by the presence of scaffolding proteins, large relay proteins to which several other relay proteins are simultaneously attached. For example, when scaffolding proteins isolated from mouse brain cells holds three protein kinases and carries these kinases with it when it binds to an appropriately activated membrane receptor. Those facilitates a specific phosphorylation cascade. In fact, researchers are finding scaffolding proteins in brain cells that permanently hold together networks of signaling pathway proteins at synapses. This hardwiring enhances the speed and accuracy of signal transfer between cells. When signaling a pathway were first discovered, they were thought to be linear, independent pathways. Our understanding of the process of cellular communication has benefited from the realization of the things are not that simple. In fact, as seen in this figure, some proteins may participate in more than one pathway, either in different cell types or in the same cell at different times or under different conditions. The view underscores the importance of permanent or transit protein complexes in the functioning of a cell. The importance of the relay proteins that serve as points of branching or intersection in signaling pathways highlighted by the problems arising when these proteins are defective or missing. For instance, in an inherited disorder called Wiskott-Aldrich syndrome, WAS, the absence of a single relay protein leads to such diverse effects as abnormal bleeding, eczema, and predisposition to infections and leukemia. These symptoms are thought to arise primarily from the absence of the protein in cells of the immune system. By studying normal cells, scientists found that the WAS protein is located just beneath the cell surface. The protein interacts both with microfilaments of the cytoskeleton and with several different components of signaling pathways that relay information from the cell surface, including pathways regulating immune cell proliferation. This multifunctional relay proteins induce both a branch point and an important intersection point in a complex signal transduction network that controls immune cell behavior. When the WAS protein is absent, the cytoskeleton is not properly organized and signaling pathways are disrupted, leading to the WAS symptoms. Termination of the signal. To keep this figure simple, we have not indicated the inactivation mechanisms that are an essential aspect of cell signaling for a cell of a multicellular organisms to remain alert and capable of responding to incoming signals. Each molecular change in its signaling pathways must last only a short time. As we saw in the cholera example, if a signaling pathway component becomes locked into one state, whether active or inactive, dire consequences for the organisms can result. Thus, a key to a cell's continuing receptiveness to regulation is the reversibility of the changes that signal produce. The binding of a signal molecules to receptors is reversible, with the result that the lower the concentration of a signal molecules, the fewer will be bound at any given moment. When signal molecules leave the receptor reverts to its inactive form. Notice that. Then by a variety of means, the relay molecules return to their inactive forms. The GTPase activity intrinsic to a G protein hydrolyzes its bound GTP. The enzyme phosphodiesterase convert CAMP to AMP protein phosphatase, inactive phosphorylated kinases, and other proteins, and so forth. As a result, the cell is soon ready to respond to a fresh signal. Okay, thanks for listening.